most of you knows me. We discussed about observed expected analysis in the first session. And now we move to discuss about another important activity of CVM, that is the cohort event monitoring. We'll be discussing with key players about the lessons learned and also about what we can improve for the future. I'll give a very quick introduction just to share with you two main considerations. One about the heterogeneity and complementarity of the data sources we can use for vaccine surveillance. And the other consideration I want to share with you is about size, about how big should be the data to support us in vaccine surveillance. I start from this. Everybody knows this definition of signal, but I want to emphasize that the multiple source part of this definition. So there is no one source fitting all the needs we have in the context of signal management. So we really need to rely on different data sources, different approaches. And I think this has been already addressed in the first part of the session this morning. And things, as you know, have been changing over time, more and more. This was a traditional approach, just a few patients studying in preclinical, in pre-marketing experimental studies, then some observational studies after that, and the spontaneous adverse report that basically can capture information from all the patients treated. But more and more, of course, we are moving towards an active monitoring environment. And I think all of these settings may contribute to generate evidence on safety of vaccines. Well, given that I like cooking, now I'll use food as a metaphor for sharing with you my idea of integration of different sources for vaccine safety surveillance. So which ingredients do we need for signal management? We start from this. This is traditional, old-fashioned pasta. I think everybody knows pasta, penne alla norma. And everybody is happy with this. So hopefully, sometimes we don't need anything else, right? So we just apply spontaneous reporting system. That's it. But a meal has to be really complete of all different ingredients, and we really need to strengthen our body. We need proteins, so we need also a nice stick, and this is, to me, is a longitudinal healthcare database, is strengthening in a way the signals. Well, where active surveillance stands, here is the salad, because we really need all sorts of ingredients. Maybe salad per se is not enough, but a meal is complete if we have also the salad. So just to give the view that all those are complementary, maybe enough per se to address some issues, but we really need to have a complete meal to be fully satisfied. Not having too much of any of this, but a bit of any of this. I hope I succeeded with these examples. And that was my first consideration about complementarity and heterogeneity. The second consideration is about data. We live in a data-hungry system. So everybody is trying to get more and more data. It seems like we are never satisfied of the quantity of data we have. And I think this was showed by Miriam this morning already. Around 2 million of reports of suspected ADR related to COVID-19 vaccines. I've never seen something like this, and hopefully we will never see something like this again in the future. But again, using the food as a metaphor, I want to share with you this consideration. This is the biggest pizza ever. This is in the Guinness, huh? And this is the biggest sushi ever. The biggest hamburger ever. And of course, because I like healthy food, this is the biggest salad ever. But pay attention, this is a study from Harvard colleagues saying that if the size of the dish would be cut by 50%, this would have dramatically dropped the prevalence of obesity in children. So let's pay attention to have a too much big data driven approach in pharmacovigilance, because it's not true we need always big data. We need thorough clinical observation. And this is a very good example. 3 reports of PML related to natalizumab led to the suspension of the drug for a while. Then we understood what was the issue underlying PML onset about the GC virus exposure, and there were some risk mitigation measures were implemented, and the drug has been reintroduced into the market, and it's still one of the most frequently used drugs for multiple sclerosis. So that were my two considerations, and now we start the session, and I have the pleasure to invite to the floor Florence van Hunsel. Florence, I think she has been already introduced. Florence is the head of the innovation of signal detection at LAREB. It was one of the key players in the context of Work Package 1 on safety surveillance in general population. Florence?
Thank you. Let's see if I get my slides. Yes, there they are. So first of all, I'm going to talk about cohort event monitoring. I'm Florence van Hunsel, but I'm also talking on behalf of my colleague Monica Raadke, who worked a lot on this cohort event monitoring for the COVID-19 vaccines and who is currently on maternity leave, so she can't be here today. And I, of course, would like to thank everybody that we worked with in the past 2 years. Before I start talking on lessons learned, I really briefly want to share some results with you to give an idea of what we did the past 2 years. And I'm doing this by sharing with you some results that we presented in Halifax at the ISPI meeting, so some of you might have heard it already. I see that's a bit wobbly. Briefly going to some results. So, it has been said a lot already that we used the COVID-19 vaccines, which were new in a very large population in multiple countries, and that really we needed to do a comprehensive safety monitoring. So what we did here was implement a cohort event monitoring study with a common protocol that we could use for real-time monitoring of the vaccines that were registered in Europe. And what we can do with this type of study is really look at real-time incidence rates of adverse events following immunization, where you also take into account the serious events or the events of special interest. So for this study, it was a prospective cohort event monitoring study rolled out in 13 countries, where we worked with 4 different methods of data collection. So the SafeFact platform in Germany, the application Open in Croatia, the LAREB intensive monitoring system, which was used by 5 countries, and Research Online platform, developed by the University of Utrecht. And we collected data on first vaccination, second vaccination, the booster. We looked at the general population, but also at special cohorts. And Nicoletta is going to tell you more about that. So in this type of study, patients register, they fill out a baseline, and then they are followed during a period of approximately 6 months. Then what type of data, what amount of data do we have? Well, we have a lot of data in here. A majority of data is collected by the German colleagues with their system. But the LAREB intensive monitoring part of the study that I'm going to be focusing on had almost 30,000 participants. And what you can then see is that there is quite a large part of the vaccinated persons who have an adverse event following immunization, but most of it is non-serious events. And especially if you look at the serious events, the frequency is very low. So what is LAREB intensive monitoring? Well, it is a system for cohort event monitoring studies that we developed at LAREB in 2006 already, and that has been implemented in a lot of studies, both for vaccines and for medicines as well. Actually, it has been evaluated This is a thesis that my colleague Linda Harmark wrote, where she really looked at all the safety studies we did with the system and did a thorough evaluation. So, for instance, we use it yearly in the Netherlands for an influenza monitor, but we also used it in the past to look at the H1N1 vaccine when we were in that pandemic. And for instance, we have a diabetes cohort that we looked at a couple of years ago. So for this study, one of the advantages was that we rolled it out in multiple countries with a common protocol. There were 5 countries using this system. We had mostly data on first and second vaccination, and we used a common data model for the analysis. So what can you then do? This is something that we're working on currently. You can pool data together and then, for instance, look at frequencies in different strata. We are looking at time to onset and time to recovery, but you can also look at covariance for developing ADRs. And this is something that we used a mixed effects model for. I'm not going into all details because we're planning a publication. But then what did we learn by doing this study? And I think this is the most important part. And I divided this in the good, the bad, the ugly. To really start with the good. I think one of the good things of having this type of study in place is that it's complementary to what you have with the spontaneous reporting system. As Gianluca has already said, it's a different type of method which can really give you insight into the safety profile of the vaccines in daily practice. And you use what you also have is that you can calculate incidence rates, frequencies with this type of study. And that is something that you cannot do with spontaneous reporting. Also, which is one of the good things, I think, is that we use patient-reported outcomes in this type of study. And as we saw in the results, there are a lot of adverse drug reactions being reported which are mild and self-limiting. And this actually gives you an idea on what type of ADRs people suffer from when they are vaccinated, what their time to onset is, their duration, the burden for patients, so you can really inform the public with these kind of results. What is also good, I think, in this study is that we were able to pool data throughout European countries and really do a common analysis. But then some of the things which are, didn't work out as much as we would have hoped. It was quite difficult in the beginning to get alignment between all of the countries. Some were already ready at the start of the rollout of the vaccination campaigns. Some countries needed more time. Alignment was quite challenging sometimes. Also, not everything about the vaccination campaigns itself was really clear at the start of vaccination. And when you have this type of study, you really work with a protocol, you schedule your questionnaires, for instance, and then when something changes in the vaccination campaigns, that is difficult to incorporate in the study. And also ethics approval in some countries took a lot of time, and there are different views in different European countries. And then for us, from the LAREB intensive monitoring side, one of the uglies is you have an enormous amount of data from Germany, but we were not able to pool that data with the other countries. So the data is there, we have published about it, but we can only use aggregated data for that analysis, and we couldn't pool it with our common data model. So some considerations. These types of studies cost money and manpower, and that is something that you really need to think about, calculate in when you're doing your evaluation. So you need to think, does this yield the information that you want, considering the cost and the manpower? I would say from our side at LAREB, I'm a bit biased. We've been doing this for a long time, and I do think the costs outweigh, the benefits outweigh the costs. Also, what was something that you need to take into account is that there were sometimes reporting obligations for countries to report data to Eurovigilance from these type of studies, and that you really need to, well, take into account that this is something that could take extra money, extra time, effort to make this happen. If you want to have this study in place, this type of study for a next pandemic, or maybe for another type of virus that is coming about, you really need to have a sustainable proposal on how you want to keep this going. And that is something that I don't have all the answers for, but I think that is something we could discuss during the panel meeting, how to do this. And also something that I'm not going to answer right away is this something that you want to have centralized throughout Europe, or is it the best way to have it in different countries and work with a common data model? So something that I really think needs further discussion. Then, if you want to see some results on the study itself, the final report has been uploaded on Zenodo, so it's available for you to look at. The data is there, and as I said, we are planning to do further publications on the results. So, with this, I would like to conclude.
Thanks a lot, Florence. Thanks also a lot for a very nice collaborative way of interacting with all of us in the context of the project. It's me? No. Okay. So the floor is open for a question. Yes, please.
If I understood it correctly, it's patient-reported outcomes, right? What about the participation rates? I would expect them to be quite low, right?
It actually depends per country, and I think Nicoletta is going to talk about that a bit more. But for us in the Netherlands, we actually had a very high participation rate from the start, because we had the momentum of the vaccination campaign just starting, and there was a lot of attention for vaccine safety. So actually the cohort was full quite quickly, and also full with elderly people, which you wouldn't maybe expect. So the participation rate, at least in the Netherlands, was okay, quite okay, but then you have to have that momentum. So if you're further on in the campaign and the vaccination rates are lower, or there's maybe less attention to safety, then you can miss that, and then, yeah, you can have lower participation rates.
Okay, so you're not inviting X number of people, you're saying to people, we have this study.
Yeah, the way it works is that people at the vaccination locations can get an invite to join the study, and they can then join online. They can fill out a baseline, enter themselves, and then they are followed through time. And then we said per vaccine we need at least 5,000 people entering the study to do some of the analyses that we planned. And once that is full, the cohort, we close it. So it's not the idea that you would follow the whole population and have, like, for instance, you say 5% of all vaccinated persons need to be in the study.
Yeah, so you're not inviting people based on anything, people are coming to you,
yeah,
so to speak.
Yeah.
Okay, thanks, interesting.
Thank you very much. Thank you very much. Do you want this? Yeah.
Oh. Thank you. Yeah, thank you very much. So I have two questions out of curiosity. The first one, you indicated that you had very few serious reports, and so the majority were non-serious. So I'm kind of curious, what did you do with the non-serious reports? Have they in any way contributed to any regulatory actions? I know you talked about the patient-reported outcomes, but I'm more curious about, what you did with that data. The second part of the question is about the German data. You also said you didn't pull that. I'm curious, why not? Thank you.
So for the first one, yes, we use patient-reported outcomes, and what you see, what we have in the study is a lot of ADRs that are already known, but also some of them were not known. So the majority of people who report something that has to do with reactogenicity, so it could be and injection site reactions. And I think what the study adds is that you really can do incidence rates of these adverse events and really give information to the public on what to expect. Because we all know that the registration studies are somewhat biased in sense of population, and with this you capture everybody who joins, so the elderly, well, all age groups, and in that way you have better information on what happens in daily practice, including time to onset, duration, how severe people themselves, or what the burden is for patients, and that is really useful info. There are some ADRs in the study which were not labeled at the point when we started, which were not labeled, and then you can use that information in signals as well. But as I said, the number of really serious events was quite low. Then for the second question, why didn't we pool the German data? Germany had already started with their own SafeVac platform in the beginning, and they were quite early, and we just couldn't pool their data in the common data model that we used for this. There might be other reasons as well. Miriam might add to this if she wants.
Well, I think what is difficult also is their methods or the protocol is slightly different, because they collect like baseline data at the end, they follow up a year, so that makes it difficult.
Thanks a lot. I think we need to go ahead. Thanks, Florence. Next speaker is Nicoletta Luxi from University of Verona. And Nicoletta was fully involved in the coordination of Work Package 2. It was monitoring and special cohorts. And I want to take the opportunity to thank all the partners that contributed to the 2 work package, but also to Joost team and Fabio. I think behind those numbers, there are thousands of TLC across all these persons. So I want to thank all of you, because it was really a tremendous amount of work. So Nicoletta.
Thank you. Thank you all. Yes, I'm speaking on behalf of Work Package 1 members, as this has been a very collaborative project with significant contribution from all the partners. And I would like to provide you with more detail about this active surveillance study of special cohort, highlighting what was the rationale for conduct this study, and to share also the main challenges and the main lessons learned from this study. The aim of the Work Package 1 was to generate, estimate, describe and compare incidence rates of patient-reported outcomes of different COVID-19 vaccines across different countries in pregnant and lactating women, children and adolescents, immunocompromised people, people with history of allergy, and people with prior SARS-CoV-2 infection, as this special cohort were excluded from pivotal trials, at least initially. So this is the timeline in general. This is the timelines at which questionnaires were sent to the vaccinees, but for some cohort we need to adapt the sequence, but also the number of questionnaires to be sent. For instance, this is an example for pregnant women, and the mailing of the questionnaire was planned based on the gestation period at the time of the recruitment, and on top of this, an end of pregnancy questionnaire was also sent 45 days after the due date to evaluate the safety outcomes in the newborn. And in addition, we also need to customize and adapt the baseline questionnaire with some additional question to better characterize those vaccines. And as you can see, we not only had different cohorts, but we had also had different countries. Meaning that the questionnaires and the web app were adapted and translated in several languages, but also we had the different monitoring at the first vaccination cycle and then the booster dose, and different contribution of the countries by dose and by cohort. So why do an active surveillance study in this context? Well, as mentioned earlier, these special cohorts were excluded, at least initially, from pivotal trials. So at the beginning of the vaccination campaign, the first safety warnings came out. So, for example, for people with history of allergy, the question was, do allergic people have a higher risk of having a severe allergic reaction? Can the type of allergy have an impact on this? So spontaneous reporting studies were conducted initially, meaning that we didn't have any incidence rate on this. The same for people with prior SARS-CoV-2 infection. We knew that those vaccines had a higher and increased reactogenicity as compared to those with no prior SARS-CoV-2 infection, but no comparison between doses or between vaccines brands were made. For immunocompromised, most of observational studies conducted initially evaluated the effectiveness instead of the safety. For pregnant and lactating women, those vaccines were excluded from, those people were excluded from pivotal trial. So concern about receiving the vaccine among pregnant and breastfeeding women arose, and so on. The same for children and adolescents. So based on Work Package 1 experience and findings, we would like to highlight what worked well and what didn't work. So what worked well? First of all, the direct involvement of vaccination centers facilitated the target inclusion of vaccines. In some countries, such as France, but also in Italy, we also had the monitors to promote the study and who also help in all the process of the registration and all the phases of the project. But this is, I think, that is the most important part. We established a very collaborative and multidisciplinary network with strong connection with the scientific community, but also with the regulatory agency. And on top of that, each country had its own network. Such an example, in Italy, we had a network with more than 30 partners throughout Italy. And from the point of view of findings, well, an added value was for sure to have a different vaccines brand, different doses, different countries compared in the same study at the same time. And on top of that, we also can compare findings from the special cohort with findings from the general population that was matched by sex, age, vaccine brand, and dose. And this made a comprehensive evaluation of the safety of COVID-19 vaccine. As an example, for people with prior SARS-CoV-2 infection, that we all knew that they had increased reactogenicity as compared to those with no prior SARS-CoV-2 infection. We can observe in our study that this is confirmed for all vaccines, Pfizer, Janssen, Moderna, except for AstraZeneca, for example, probably due to the fact that AstraZeneca had an increased reactogenicity %. And the same goes for the second and the booster dose. So what didn't work? First of all, having a tool ready and working and easy to adapt is crucial for a timely start of the study. Because the recruitment of subjects was not equally successful in all countries. And if we had a look at the Netherlands, for example, the Netherlands had the tool ready, started earlier, and recruited the most of vaccines. For the other countries, for example, we need to adapt, translate the tool and the questionnaire. And this is strictly related to the fact that it is crucial to start the study at the time of the start of the vaccination campaign as well, because this may have an impact on cohorts. For example, immunocompromised were prioritized during the vaccination campaign, and again, the Netherlands was the country that recruited most. And if you have a look also at pregnant and lactating women cohorts, the recruitment did not go well. Maybe to the fact that we have not enough countries participating to this cohort, but also this is related to the fact that the recruitment starts too late due to ethical committee procedures. So maybe for this cohort, but also for children, it might be useful to involve specific networks like pediatrician or gynecologist to ensure a greater involvement of these vaccines. For the booster, well, the recruitment went a little better, yeah. But of course, for the booster, we had the questionnaires ready, but of note in these cases, as said before, as Florence said before, we have another tool. During the study, we had to switch from a tool to another for the booster monitoring. So this also may have an impact on this. And this is an example of vaccines recruited in Italy in relation to the vaccination campaign in Italy. And if we have a look at the start, okay. We started the study here, and this is the vaccination campaign that was already advanced in Italy. So we can see a peak for the first dose only in this part that is related to the authorization of COVID-19 vaccine in children. And again, the booster, the recruitment of vaccine as booster with the vaccination campaign was, well, satisfactory, let's say. The second point, the loss to follow-up. In general, we will include in the future studies vaccines with the baseline questionnaire and the follow-up, and at least one follow-up questionnaire, meaning that, if you look at this slide, that we are missing the 15–30% of vaccines recruiting at baseline following the first and the second and the booster dose. So maybe this in some cases could be physiological, but we observed a very low percentage of serious adverse events that could have been prevented vaccines from filling the follow-up questionnaire. So maybe it could be useful implement a strategy to intensify the follow-up and to involve also the clinicians that work in the setting of care. And what about the extra question? We spent a lot of effort to customize the questionnaire with the extra question to better characterize those vaccines. But how many of these extra questions were actually filled? So this is an example of questions for people with the history of allergy. We know that almost of those vaccines completed the reported information about the type of allergy that they had, but what about the other question? So maybe it could be useful also in this case to have clinicians ready to support in all the phases, also in the filling the questionnaire. So in conclusion, active surveillance studies in special cohort may provide additional safety information, and it's crucial to have a tool ready, working and easily to adapt for a rapid response, and maybe a patient-reported outcome study per se, it might be not enough. Maybe it is worth considering to have a patient-assisted outcome study. And the involvement of clinicians may increase the quantity and the quality also of the information collected and can ensure a greater response. Let me conclude with thank you all, Miriam, first of all, the colleagues from LAREB and UMCU, not only for developing the tool, but also for the collaboration. Fabio from Team IT, Professor Gianluca Trifirò, and my colleagues Chiara and Francesco, and all the Work Package 1 members. Thank you all.
Thank you, Nicoletta. Don't run away. We have time for a question. Yes, two questions.
Did your questions also cover stuff like unexpected bleeding, irregular menstruation, chronic fatigue, cognitive difficulties, a lot of the stuff that the national competent authorities are struggling with providing answers to?
Well, I was also involved in the coding process of the questionnaire that we received. And I can tell you that from our side, from Italy perspective, but also from the other countries, we didn't observe many of these outcomes. So this is...
And the list of solicited adverse events was defined a priori. So, of course, some new potential signals came out later on, but still there was a chance to report these unsolicited adverse events. But actually, we didn't observe so many of these.
We've had this issue with HPV vaccination, with these CFS-ME-like symptoms, and we are also hearing stories now about that in response to the COVID-19 vaccines. So it would be nice if there were cohort studies with patient-reported outcomes that could address these questions. So we have some evidence-based answers.
Potentially all adverse reactions, suspected adverse reactions, were reported by vaccinists, but of course the list of solicited, the ones that were explicitly mentioned, was defined a priori and agreed also with the EMA initially. So of course we cannot adjust over time, or maybe yes, I don't know, it's something we may discuss later on in the panel discussion. Thanks.
So in one of your slides, you mentioned that there is like a big difference between countries in the participations in different cohorts. Yeah, so besides the issues of translating and adapting the tools, are there any barriers for the countries to participating in different cohorts?
Well, the point is that we adapt the questionnaire. Then we invite all the other countries, and not all countries would like to participate to all the cohort, also based on their vaccination campaign at the moment of the start of the study. And they had to translate and adapt also in terms of content, the questionnaire. But I think that this was not a barrier.
Hi, Nicoletta. Thank you for the presentation. I want to add something in terms of logistic, how we recruit participants in Spain. In IDEA was participating in the study as well. It was a very challenging study, since we need to first adapt the questionnaire, and not just in Spanish. Also, we adapt the questionnaire culturally. All the questions were adapted to the Catalan cultural and everything. One limitation was that the questionnaire were not translated to Catalan. So that was a limitation from our side, that people who speak Catalan was not included in this cohort. And also regarding to the follow-up, also we need to include more nurses to follow all the patients, because I think compared to other countries, we invite all the participants, because it was a challenge just to put all the information in the primary healthcare centers. Nobody wants to do that. So in the primary care centers, all the healthcare professionals invited all the people who participate in this study, and then go to the primary care center for vaccination. And also one limitation from our side is that at least in our region, we were not allowed to promote the study in the massive centers. So that was another limitation from our side to recruit participants.
Let's not spoil all the points for the discussion, for the panel discussion. So thanks, Nicoletta. We have 10 minutes extra as a gift from the coordinator for the panel discussion. So I would like to invite. Thank you again. I would like to invite Catherine Cohet. You already know. Catherine, please take a seat. Nicolas Thurin from Bordeaux team, another key researcher in active surveillance. And Joost. Joost, okay. Okay, so basically here we have the regulator, the researcher, and the IT. So we have all the components that played a key role in the active surveillance. And I'd like to ask, first of all, to all of you, what is the additional value that you really documented in performing active surveillance? I'd like to start from Nicolas, and I would like to ask you to convince Catherine to fund again an active surveillance studies to this network for the next pandemic. So what are the arguments you would like to use?
Thank you. It's not a tough task you give me. So actually, I think that there are two perspectives. The first one will be to start by the PV from pharmacovigilance. So what it brings is that now we have a denominators, because we speak earlier about the denominators when you do observed to expected analysis. I mean, it's the same here. We'll have the patient-reported outcomes and the denominator, so it's added value to quantify the risk. And then from the pharmacoepidemiology perspective, which is great, is that you can capture outcomes that are not captured in the database, in the medico-administrative database. All the non-severe cases that most of the claims data cannot capture. But you also have the chance to follow patients, in live. It's very short. You don't have to wait for the lag of the different medico-administrative database to capture the outcome. So for me, it's really something great.
Catherine, are you convinced?
I'm quite convinced. I'm really not good with those things today. No, I'm actually quite convinced, and your first argument was my first as well, which is to have a denominator and to be able to generate incidence rates for those events. Another added value, I think, so you mentioned that you can capture data which is not, that you cannot get from healthcare databases, but you can also capture data that you cannot get from spontaneous reporting systems, because since you are speaking directly to patients, you can elaborate a little bit on potential confounders. Then there is one other point which is more, I think, from the communication perspective. So there was a lot of discussion about whether all those studies are useful for PRAC, for EMA committees, etc. But it's also useful from a regulatory communication perspective, because it shows that there are actually actions taken to monitor those vaccines. And I'll see also a benefit for the people who are involved in those studies. It's like a direct personal contribution, and it's also a way to show that the general public is involved. And maybe it's taking it a bit too far, but we could think that it's the type of work that could be used against anti-vax movements and all that. Maybe that's a bit idealistic.
Okay. Thank you, Catherine. Now we have the funding, but without the tool, we cannot go ahead. So we'd like to hear from Joost. What are your perceptions about the value of what we've been performing in the last years?
So I can, from IT perspective, what we have added the key are 4 things I think that are necessary to be able to do this. So the first of all is have a group of people with knowledge. So I have to thank my team, also Ellen van Deuren and Mark Padrosa over there, with a lot of effort having this done, be able to set this up, have the knowledge and the time to work on this. But the second thing is the system itself to do the data entry. So we are lucky enough to have a really flexible data collection system in place that is able to have the possibility to add categories, to add other workflows, data flows. And we are very happy with the network of Miriam, of course, that we could copy the whole process from the LAREB. So we have the questionnaires already, we have the workflow already. So that's really key to have this network in place and share this knowledge together. In that way, we could easily speed up to create this data collection module of our own. And the third and the fourth thing is to have the data stream pipeline at the end, to have this in automated process to the people that are working with the data. So that are the 4 key elements that really helped in speeding up this process and have this data collection ready in time.
Thank you, Joost. So before leaving space to the audience, I would like to ask you also what didn't work and what may be the solutions to be implemented eventually in the future to prevent mistakes or challenges we encountered during the activities we have carried out. So let's have another cycle. Let's start from Nicolas.
Me again. So I think the key words, Nicoletta said it, it's just like anticipation, to have everything ready, including the funding. Because, for example, in France, that was the main challenge, because we had funding from the EMA, but unfortunately it was not enough to run everything properly, so we had to ask a complementary funding from the French Ministry of Health, which was quite challenging to start on time, so we missed the momentum, as it was previously explained, and that implied that we had to recruit a lot of manpower internally to go in the different vaccination center, to promote recruitment, so it was quite a challenge. So I will say that funding and anticipation.
Catherine?
I think in terms of the challenges, I'm really more looking to the future, which is how can those tremendous efforts being reused, and how can we be better prepared for the next pandemic, because such a network is, I don't know to which extent it's sustainable. You are the people who are in the driving seat. So I think it's about sustainability and being able to respond faster. Another challenge, I guess, and I didn't think about it, but it's true, Anders asked the question about what was the response rate, and from a selection bias point of view, it's interesting. Like in clinical trials, we have like screening logs where we go to people and then we are able to calculate the response rate. So I don't know, there might be issues with selection bias, but overall, yeah, I think it's really maintaining such a network or being able to restart it very quickly.
If I can just add something about the selection bias, actually the issue we had here is that we were not the master of the recruitment, because we start late. If you start earlier, you can choose to include, 10% of the person that are visiting you in the first month, and to repeat it every month to be able to capture every single slide of the population covered by the recommendation.
And can I just, one quick additional things in terms of the challenge that we encountered is there were many questions to EMA in the beginning about the impact on reporting to national pharmacovigilance system. So how could we reconcile this, and that it was a
to
challenge.
comment on this, well, here there are Chiara and Francesco were monitor in the vaccination center, but they can document this. Well, it was almost risky to try to engage vaccinees when we started with the recruitment, because they were the reluctant that were obliged to get vaccinated. So I think even was kind of risky to talk to them about potential safety issues concerning vaccines. So I think, I hope it's something that won't occur again in the future, but really that was an extreme selection bias. So we were selecting the ones really that didn't want to participate at all. So it was really an issue.
Just something we missed from the beginning, but I think that communication is very, very important. Because when we start with the first poster provided by LAREB and the team, which looked quite nice to us, but we had some comment from physicians saying, but what did you put the word safer on it? Doesn't it safe enough? And so we really have to think truly how we will communicate, how we have to communicate with patients.
And I think communication is important also between IT and researchers, because I mean, we encounter some issues in such a short time in incorporating all the elements that we like to collect and translate these into IT tools. So I would like to hear also from you, what do you think could be improved for the future, and well, what do you think about this challenge of communication between researchers?
Yeah, so I think it is for all our projects that we are running, it's always communication, and when you're working in a complex environment with different groups of expertise, that's the responsibilities and definitions is always key elements to have a lot of attention in the beginning of the project that will prevent errors later on or misunderstandings. So I think that's always the good thing we all know that, but it's really important to have that specified at the beginning of a project and have regular meetings and discuss this and what goes wrong and that you are on top of it.
Yeah, on top of communication, I would say it's important also trusting each other, because there was a kind of frustration from researcher as not having available the tool, and there was an issue in defining together what would be the right timelines, so there were expectations. So I feel this is also very important to create trust networks of people.
Yeah, so that's of course we are the first time working together. So when you have multiple projects, you better learn each other, and then you know how to trust each other. That's of course.
Okay, I would like to hear now from the audience. I see Rosa, Agnes. I cannot tell you who was first, so you tell me who was first. Agnes?
Yeah, thank you. Well, first of all, the communication is very important, and the outcomes of this cohort event monitoring for the communication in our country was very important. So I totally agree with you. Maybe you don't see that much severe adverse events, but how many fever are to be expected, and are there more fever expected by the one vaccine compared to the other one? For instance, RGP wants to vaccinate all their personnel. Don't do that in 1 week, because then you have all of them out of office. So that kind of information is for society quite important. But the other thing I want to ask also is, if we want to do this again in the future, we have a lot of lessons learned, but how can we make this infrastructure sustainable? Because, to be honest, we have the infrastructure for our country, but the European network, and to roll it out very fast in also other countries, that needs a sustainable network, and that's not going to be for free, to be honest. So that's a question maybe to the EMA or to all of you that have thoughts about this. And I have one very specific question to Catherine. Sorry, but and that's about the reports going to vigilance, because we had a lot of struggle with that one. That costs us a lot of efforts, costs us a lot of time. We did an automation on this, but what is the usefulness to also send non-severe events to UDA? Because we were obliged to, but we didn't think it's not that useful. That's useful for the other purpose, for the communication, but not for the signal detection in UDA vigilance. So in the future, please just let us be the severe to send to you, because that's gonna, it's a burden. Yeah.
Yes, no, and it's a burden on the ultra-vigilance side as well, obviously. So, no, I mean, I don't have the answer, but I cannot not agree. Now, your first comment about sustainability and having pan-European systems in place, and I don't have an answer either, but I guess maybe a start could be thinking of having such networks in place for not necessarily for pandemic purposes. There are vaccines which also require monitoring, and again thinking of RSV, my favorite at the moment. So I mean, flu, there are still some potential issues with flu. So obviously it's a question of funding, of infrastructure, etc. But maybe going in a more inter-pandemic strategies, and that would obviously would have to be taken at European level, and with the new regulation, European Health Union and all that, maybe it's something that could come on the table. But that's very tentative from my side.
Just let me say that Andrea Farkas from Romania was supposed to be here just to exactly discuss on sustainability also from Eastern countries, because we are used to have infrastructure readily available or funding in Western countries, but Eastern countries have different issues, and that was one of the points that she raised. Rosa and Miriam. Miriam, you want to comment on this, and then Rosa, or Rosa first? Rosa first. Yes.
I was wondering so you are speaking about recruitment at vaccination centers, but at the time there was, especially at the beginning, there was awareness, there was probably there were people that would have been eager to register for the study before vaccination, which would have allowed other study designs to be enacted and having a kind of background rates or something or whatever. So and really also probably enlarge the recruitment, give yourself more opportunities to recruit. Is there any comment on that?
Well, from a technical perspective, there was an issue with informed consent, because we wanted to have access to vaccination. There were a list of people to vaccinate at the beginning, especially immunocompromised. So we explored this, but there was an issue also in terms of informed consent. So we need to have informed consent for patients to contact them. So we couldn't ask the list of the ones that were supposed to get vaccinated, because.
No, but I'm speaking about I did promote some advertisement that you gave me, and there were people who would have submitted voluntarily, entered the website in that moment spontaneously.
We disseminated as much as we could, but of course, I mean, it's not an easy task. We involved in Italy 30 partners, but of course, you really need to have monitored there daily, convincing people and supporting people in getting registered. So I think that's the most effective way to recruit people. I think also France have the same experience. I think it was Miriam.
Well, I think we need to wrap up this session, but I have a thought, which thank you, Anders, for your comments, because what I'm actually thinking now is, like, if you want to have such a system in place, and again, you can include many Eastern European countries which are not those that have large linked databases at the moment yet, maybe so much, but should we ask different questions than the reactogenicity questions, because those you actually get from the trial. So should we ask for especially those syndromic or the ones we struggle with most a priori, or menstrual disorders, or things that may be not typical, but we have seen now several times, that we put those on the solicited list rather than the fever and the injection site reactions and etc.
Yeah, no, it's true. With also the potential limitations that some of those are also extremely rare. But yes, why not?
I'm looking at Miriam to understand if there is time for. There is Xavier, and there are two colleagues that raised. Okay.
Do we have time?
Yes, Xavier.
No, I just have a comment, because you talked about communication and the need for communication. I would like to say that from the EMA side, perhaps unexpectedly, there has been the need for communicating towards the committees, actually scientific committees, because it was sometimes considered that EMA wanted to implement and establish an alternative system of pharmacovigilance. And it needed to be explained that it is not to replace pharmacovigilance, and probably this need also to be explained to the healthcare professionals and patients. And this is an additional way of collecting information with different objectives. And I think this is very important to realize that in terms, this is not an alternative way of. This is not a replacement of the routine pharmacovigilance system, which could be perhaps misinterpreted.
Yeah, thanks for this very, very, very last quick comment from the colleague here up front. Yes.
Thank you. I'm Camelia Bușa from Romania, actually.
Okay.
Yeah, and I just wanted to give a thought regarding maybe using this system that we already have in place for addressing vaccine hesitancy. Because in my country, we found a lot of people being impressed that authorities are investigating adverse effects of the vaccines. Of course, they don't know what is happening before the authorization, but they see that this is a way of authorities' surveillance of the vaccines. So maybe this can be a joint aim.
Yeah, again, it's a risk communication issue. I don't know, Miriam, if you want to wrap up, or we just close the session like this. So thanks everybody, thanks the panelists, the speakers, and have lunch.